Good afternoon, everybody. I am talking about proteomics and lipidomics in heart failure. To provide an outline for the presentation, first I will define proteomics and lipidomics, and to then introduce new areas of lipidomics and proteomics. I will first talk about cardiac energy metabolism, oxidative phosphorylation, the formation of reactive oxygen species, the consequences of oxidative stress, and the new areas of Stress Proteomics and Oxidative Lipidomics. I'll conclude with some preliminary results using lipidomics in heart failure that's initiated at QCRC. So in the study of an organism's genotype and phenotype, we look at DNA, RNA, protein, and metabolite. So after the studies of genomics and transcriptomics, we have the areas of proteomics and metabolomics. Proteomics studies the proteome, which is all proteins expressed by an organism, and to study the proteome, we use chromatographic equipment such as liquid chromatography, mass spectrometry, as well as 2D gel electrophoresis systems. The results are shown as protein profiles, where we can look at up and down regulated proteins and compare between different experimental conditions. Metabolomics, on the other hand, is the study of the metabolome, which is the complete set of small molecule chemicals found in an organism. They can include, for example, amino acids, organic acids, sugars, vitamins, and fatty acids. Due to the vast diversity of the chemical properties of these metabolites, there is no one analytical platform which can comprehensively cover the entire metabolome. Nevertheless, we do attempt to do that with several methods in using gas chromatography and liquid chromatography and with tandem mass spectrometry. So some of the subset fields that have come from metabolomics are, for example, glycomics, which is the study of all sugars and carbohydrates, and lipidomics, which is the study of all lipids. Results are once again shown as metabolite profiles, and we use multivariate data analysis to differentiate between our sample groups. To understand the consequences of oxidative stress in the heart, first let's look at normal cardiac energy metabolism. The heart produces energy in the mitochondrion, of which there are several thousands in one individual cardiomyocyte. In the fed state, the heart normally uses free fatty acids, and 90% of energy comes from the catabolism of free fatty acids. Free fatty acids are converted to acetyl-CoA through beta oxidation and then enter the Krebs cycle. Glucose, on the other hand, undergoes glycolysis, gets converted to pyruvate, enters the mitochondrion, gets converted to acetyl-CoA as well, and then goes through the Krebs cycle. The heart has very high energy demands and very little energy storage capacity, and therefore these energy metabolic pathways need to be very dynamic in order to respond to the heart's needs. ATP, or adenosine triphosphate, is the main energy source for the majority of cellular functions, and through the metabolic pathways that I've shown so far, there's only very minimal ATP that's produced through the glycolysis pathway. So this ATP is not sufficient for normal cardiac function. However, what's interesting is that all of this process so far, it has not used oxygen at all. And at the same time, there are a number of reduced cofactors that get produced through these processes. These reduced cofactors, such as NADH and FADH2, they drive a process called oxidative phosphorylation. And this utilizes oxygen and occurs at the inner mitochondrial membrane. So if we look at the inner mitochondrial membrane, there are a number of proteins that are involved with oxidative phosphorylation. NADH and FADH2 are able to donate their electrons to these protein complexes, and it drives a series of redox reactions across these proteins. At the same time, electrons are passed through these proteins through what is called the electron transport chain. The final electron that comes out of protein complex 4 is accepted by oxygen, gets converted to water, and this, at the same time, this protein complex is able to pump protons into the inner mitochondrial space. This develops a hydrogen gradient across the inner mitochondrial membrane, and this gradient assists protein complex 5 to phosphorylate adenosine diphosphate to adenosine triphosphate, thereby making the required energy for the heart. Now, it does occasionally occur in normal oxidative phosphorylation that there is some electron leakage from these protein complexes, and what happens in that situation is that oxygen comes in and accepts those electrons, making a superoxide ion. This superoxide ion is a member of the reactive oxygen species, and the body is capable of taking care of the danger from the reactive oxygen species. How that happens is that the superoxide ion is converted to hydrogen peroxide and then onto water and oxygen through naturally occurring enzymes of the body. These enzymes are believed to be located very close to the location of oxidative phosphorylation, so that very minimal damage takes place from these reactive oxygen species, in particular hydrogen peroxide, which is quite dangerous. So what happens in the situation in some cases of heart failure where the cardiac muscle is not being supplied with the sufficient oxygen required for its function? Well, what happens is that oxygen is not here or is not present at the end of the electron transport chain to accept the electron coming from protein complex 4, which means that the proton gradient is not being established correctly. This is not able to drive protein complex 5, which means that there is a decreased amount of ATP that is able to be produced. At the same time, this electron transport chain stops functioning and might even go in reverse, which means there is increased electron leakage. These electrons are converted to hydrogen peroxide, and the concentration of hydrogen peroxide increases dramatically in the mitochondrion. At some point, the capability of the naturally occurring enzymes to cope with this increased level of hydrogen peroxide will end, and the hydrogen peroxide is then does react with ferrous and ferric ions to produce hydroxy radical and hydroxyperoxyl radicals. And these radicals are incredibly reactive members of the oxygen species, and they will damage anything and everything that is surrounding this region. And this usually means damage to the membrane, cell membranes, as well as nearby proteins. These radicals are also able to diffuse through the membrane and attack other parts of the cell. Now, usually, if the amount of damage is severe, this cell will undergo apoptosis, and if this happens continually, it means that there is cardiac damage and therefore heart failure. So, how the excess of reactive oxygen species causes DNA damage, protein damage, and lipid damage? How do we study the reactive oxygen species? Well, we can't measure reactive oxygen species because they have a very short half-life and they react with everything. And so in the process of extracting them, we will not have the oxygen species as they are, which means that we then need to measure the protein and lipid damage that comes from having had these reactive oxygen species present. So one of the new areas of proteomics is called Stress Proteomics. It aims to identify and quantify redox-based changes within the proteome under conditions of oxidative stress. Here is one study related to heart failure where we are looking at the concentrations of protein carbonyls, which is a modification caused by several series of reactions from reactive oxygen species. The scientists here extracted the proteome from post-mortem heart tissue and discovered that protein carbonyls were much higher in heart failure patients than in control patients. Oxidative lipidomics is the identification and structural characterization of lipids and oxidized lipids in cells, tissues, and biofluids. One particular lipid which is highly prone to damage by reactive oxygen species is cardiolipin. It's a unique phospholipid in the inner mitochondrial membrane only, and it has an essential role in mediating mitochondrial oxidative metabolism. Cardiolipin is particularly susceptible to oxidation because it exists in this form where it has 4 fatty acids with 2 double bonds each. Now, lipid peroxidation occurs at the site of double bonds in the fatty acid chain, and lipid peroxidation changes the ability of cardiolipin to maintain membrane permeability. So, in heart failure, it has been found that there is a decline in cardiolipin content in the inner mitochondrial membrane, and that cardiolipin oxidation decreases mitochondrial oxidative metabolism. Recent papers have also found that there are new techniques to actually measure the oxidized fatty acids using more complicated techniques. And so I'm sure in the future, there were no papers on heart failure related to this, but I'm sure in the future it is just a matter of time before a publication comes forward with this kind of analysis. Now I will talk about an ongoing study at QCRC, and where we are using lipidomics to look at our samples from patients. The idea is to find a lipid biomarker for an unstable coronary plaque. As you know, heart failure can sometimes be caused by thrombosis that occurs in the coronary arteries, blocking blood supply to the cardiac muscles, and thereby necrosis occurs and heart failure then follows. So we were fortunate at QCRC to obtain 9 unstable angina blood plasma samples and 8 ST-elevated myocardial infarct samples. These are some of the patient characteristics, showing no significant difference between the lipid profiles that we normally find from hospital tests. This slide will, through this slide, I will describe very briefly how a lipidomic study takes place. So first, we collect the sample, the blood sample, we centrifuge, and we keep the plasma. We store the plasma in -80 until we're ready to analyze. On the day of analysis, we use a liquid-liquid extraction process where we try to get the lipids to transfer from the aqueous phase into the organic phase, and we use chloroform and methanol to do this. Once the lipids have transferred into the organic phase, we collect the organic phase, dry it down, and reconstitute it in an appropriate solvent for injection into a liquid chromatography system. So here is an image of a typical LC-MS system that we use in the laboratory. And here is a schematic of the mass spectrometer. It's called a Q-Exactive instrument, and we have this at QCRC. The eluent from the chromatographic column enters at this point in the mass spectrometer, where it undergoes electrospray ionization. And we use polarity switching at this point to ionize the molecules in negative and positive mode in order to get a comprehensive coverage of lipid classes, because there are some lipids which will ionize preferentially in the positive mode rather than the negative mode, and vice versa. The lipids travel through into the first section of the mass spectrometer, where the entire mass of the lipid is measured. It then fragments into characteristically according to lipid class in the second section, and these fragments are measured with very high accuracy in the Orbitrap section. Here are typical chromatograms that we see in positive mode and negative mode. This is a 90-minute run per subject sample, and these peaks are lipids that are eluting from the column and originating from the blood sample. Certain classes of lipids will elute at certain regions of this chromatogram due to their hydrophobicity. Lipid identification occurs by piecing together information from the entire mass of the lipid as well as its individual fragments. Through this process, we are able to not only tell what lipid class it is, but we are also able to tell how many carbons there are in each fatty acid and how many double bonds there are. We use a database called Lipid Search in order to confirm our identities, and we use Progenesis QI to do statistical analysis and multivariate data analysis. These are results from one particular subject. This is an unstable angina patient. The top 3 lipid classes that we see in this patient's blood is phosphatidylcholine, triglyceride, and sphingomyelin. We were able to identify 288 different types of phosphatidylcholine. Phosphatidylcholine is a cell membrane lipid, and in this particular form is the most dominant form that I was able to detect. Triglyceride is an energy storage molecule, and this is the most predominant triglyceride we found. We could see 188 different types of triglycerides that we could identify. Sphingomyelin is also found in the cell membrane and is also a signaling molecule. There were 423 other types of lipids as well in this patient's blood sample that we could identify. If we look at the total number of compounds across all subjects, we were able to see more than 26,000 compounds in positive ion mode and 13,000 compounds in negative ion mode. The number of compounds we could identify were more than 1,600 in positive and 874 in negative mode. If we look at the statistical significance of these results between our unstable angina and STEMI groups, there were 8 compounds in positive and 8 compounds in negative mode that showed a significant difference of with a p-value less than 0.001. We are yet to identify what those lipids are. In the meantime, our multivariate data analysis shows mixed clustering at this point between our STEMI and unstable angina groups. The blue dots represent subject samples, and the purple dots are unstable angina. We hope that as we add more subject results to this collection of samples, data, that we will be able to find differentiation between the STEMI and unstable angina groups. To conclude, the emerging areas of stress proteomics and oxidative lipidomics hold great promise in the study of oxidative stress in heart failure, and QCRC is striving to aid healthcare professionals study the cause and treatment of cardiac disease, as well as developing risk stratification tools for patient care in Qatar and beyond. And I really hope proteomics and lipidomics will make a significant impact on that. Finally, I'd like to thank members of QCRC, Thermo Scientific, Nonlinear Dynamics, the Heart Hospital, and Qatar University. Thank you.